A very good morning, one and all, and a warm welcome to all the participants, especially the student community, to the webinar on microbiomes. No organism is an island. This webinar is exclusively organized for the students. First ever webinar from KE organized exclusively for students. So I think this is a great opportunity for all the student community to hear from a renowned microbiologist from IARI. So on behalf of Marburg Institute of Agriculture, I wholeheartedly welcome Dr. B Ramakrishnan, Principal Scientist, Division of Microbiology, ICR, Indian Agricultural Research Institute, New Delhi. Now I hand over the floor to Dr. Iniya Kumar, Assistant Professor, Agricultural Microbiology, Marburg Institute of Agriculture, and he's the organizer of this webinar to introduce our guest. Sir, please.
Morning, all of you. Thank you very much for joining us. It gives me immense pleasure to introduce guest speaker today. Dr. B Ramakrishnan was an alumni of TNAU. He earned his bachelor's degree from Tamil Nadu Agriculture University, and then his master's was in Professor B.N. Jory's lab at G.P. Pant University of Agriculture and Technology. His doctorate in microbiology was from the laboratory of Professor K.V.B.R. Tilak, a renowned national level microbiologist in Indian Agriculture Research Institute, New Delhi. After completion of his PhD, he joined Agricultural Research Service in ICR and worked under Dr. N. Sethuraman of Central Rice Research Institute, Cuttack, Odisha, from 1993 to 2010. He had his sabbatical to pursue a part of his research in Max-Planck Institute of Terrestrial Microbiology, Germany, by BOYSCAST fellowship from ST. That he also a Fulbright fellow under the leadership of Professor Derek Lee, University of Massachusetts. He was also a holder of a research with Professor Karaj, Professor Ravi Naidu, University of Adelaide. So it gives me an utmost pleasure to welcoming our guest speaker of today to give a talk on microbiomes. Microbiomes, as you all know, is an interesting topic. I would like to invite Dr. B Ramakrishnan to give his talk. Please, sir.
Yeah. Good morning. Dr. Iniya Kumar and students who are interested in microbiome and microbiology. And it's my pleasure to give a talk on microbiomes. Am I audible?
Yes, sir. Yes, sir.
Yes, sir. Okay, so the topic is on microbiomes, and so I wanted to give an emphasis that no organism is an island. In fact, like I gave this topic from inspired from the poem of John Donne, who said in 1624, "No man is an island, entire of itself, and each is a piece of the continent, a part of the main." And again, he went on to say that therefore said not to know from whom the bell tolls, it tolls for thee. It actually expresses that idea that human beings do badly when isolated from others and need to be a part of a community in order to thrive. So this is a way we could understand about the human life in our society. And also I wanted to give an impression, or rather the new ideas, a new perspective on organisms, on all the living organisms on the earth, which is not an island itself. And I just want to take you to important questions. We are all concerned about the philosophical question as well as scientific question, who we are and who are you. These two are often, we ask ourselves. There is an interesting website, which is by BBC, British Broadcasting Corporation, where they say how much of your body is your own. And in that website, you can enter your date of birth, sex, and choose a sex, and also use a units, height and weight. And if you go say go, then you will have information about the body shape, body count, body size, and body builder. So what are the information you're going to get? It will say more about the ingredients of yourself, how much of oxygen, carbon, hydrogen, nitrogen, calcium, and other elements which are present in you. And also like how much you could dig for the trace elements like arsenic, mercury, and gold and silver. And then gives you like number of atoms present in you for your body size and body weight and body and height. And interestingly, they also give you the total price tag in terms of dollars, 1,897 for all the elements which are present in you. And then they also give you the in terms of numbers. What is the number of red blood cells, platelets, and then the and then skin cells and other things. There is something which is interesting at the bottom, where they will say about fellow travelers. How many microbiomes or microbes am I carrying? So they say about 100 trillion microorganisms you are carrying in yourself. And how much of this, these microbes weigh? So, it's almost closer to your brain. Brain, if you say 1.4 kg in your body weight, and it is microbes will almost weigh about 1.3 kg. They also give you the number of genes. And if you look at it, like you have only 20,000 genes, human genes, but the microorganisms which are associated with you, they carry about 2 million genes. This is what they say about you. And also they give you information about the body size and the body builder in terms of, how many times your body clocks, how many billion times a heartbeat, and other bodily functions and other things. And which is very interesting website for you to understand about the life as such, like we undergo, or rather we feel in the earth as a human being. And finally, they give you the vital statistics where they say about how much of DNA you have. I have something around 800 MB, and then the price tag is about $1,897 in terms of elements which are present in my body. And also it will say about how many cells I have. I have something around 32.5 trillion, 32.5 into 10 to the power 12 cells I have. And also I have about 100 trillion of microorganisms in my body. And then they also give you the number of atoms which are present in your body. This is something very interesting. We have, when you talk about microorganism on your human body, we have something closer to 39 trillion microbial cells, which is almost like 1 to 3% of the total biomass. And then that is like human body is teeming with bacteria, fungi, viruses, and other microorganisms. And most of these functions are associated, still not known. So that is very interesting. So if you go by the number of cells, as I told you, like when you talk about human cells, if you say 10 to the power 13, and then roughly 10 to the power 14 microbial cells are present on the human body. Also, interestingly, every gram of fecal material will have something around 10 to the power 12 bacteriophages, while they have about 10 to the power 11 bacteria per gram of the fecal material. So this is the kind of size of the different entities which are present in the human body. That's really interesting. But of course, these estimates are getting revised now. And there is a paper by Sander, which was published in PLAS Biology, where he said like human cells, if they are about 30 into 10 to the power 12 trillion cells, and bacteria will be around 38 trillion cells. Of course, you could also say in terms of mass, those information is also given. But what is interesting is like the bacteria to human cells, which is also like, they differ between a man and woman, and a young infant and elder and obese people. So that means like there is some kind of an association is always with microorganisms by the human beings. So finally, if you're talking about a human being, you are not just one single organism. You are a superorganism. You are a single organism that is made up of billions of other organisms. Of course, all these organisms could be bacteria, fungi, viruses, and other things. This is what we call them as microbiome. And there are about like 500 to 1,000 species found on the human body, and which is very important for human health and even the brain function. So I'll take you back to the cells which have been described by us. So if you look at it, like, we know bacterial cells are typically will range in the range of 1 micrometer, whereas the yeast cell something around 5 micrometer, whereas the animal cell will be around 10 micrometer, but each one of them has got a different kind of properties. But if you look at them, like, they will have what you call a number of proteins is 3 into 10 to the power 6, whereas yeast will have about 10 to the power 8 to the power 8, whereas mammalians will have about 10 to the power 10 number of protein molecules. And also they have something like, membrane proteins, which will differ by, twofold. I mean, 10 to the power 5, 10 to the power 7, 10 to the power 9. And then we have DNA, which is like 5 to 10 to the power 6 base pairs of DNA, which is present in the bacterium, whereas about one-fold increase, like 1.2 into 10 to the power 7 base pairs, which is present in the yeast, and 3 into 10 to the power 9 base pair is present in the human cells, mammalian cell. And like it is not, when you look at these cells, you need to know, you need to believe yourself like you are looking at something which is, cell. So we have a limited capacity to see them because our eye has got a limited resolution power. So we could at the maximum see something around 1 millimeter or maybe less than that. But you need to have someone, like some aid. So earlier days, the light microscope was used. Then the electron microscopes are used. So I will give you some understanding about the microscopy, how these developments help to build the new concepts in life, and about the microorganisms associated with present in the earth in different niches and different environments. So this is a kind of cartoon to show you the different cell size and shape of microorganisms which are now recorded or have been observed by several scientists. So you see, you can name anything like these cells are something very interesting because none of them are by any kind of what you call symmetrical shapes. You got all kinds of size and shapes with respect to microorganism. Each one has been reported in the sense of like, they could also be seen by the name, like genus name, genera, and species, which is available in a paper which is published in 2006 by Young in Microbiology and Molecular Biology Review. That's very interesting. So I'm just giving you a picture where you would be able to appreciate the microscopic molecules of microorganisms and make some comparison between them. So if you look at a bacterium, which is about 1 micrometer or 1000 nanometer with 3000 nanometer of length, if you compare with that, red blood cells will be about 10 micrometer. And of course, we have bacteriophages and smallpox and viruses, which are very, very small. So there are, you have not only molecules and microorganisms of different size and shapes, which are available. The next question comes, like we call several organisms a microorganism. So how big a microorganism you can find in the nature? That is something which is an interesting question for many of us because we need to know like whether we will be able to see them without the aid of a microscope. Of course, there is one organism called Margareta nambiensis, which was discovered in ozonic sediments of Nambian coast in 1997. And that is currently holding the world record, like something which we all understand by Guinness World Record. The similar way, this world record for the largest known bacterium is still be with this organism, Margareta nambiensis. This microbes ranges from 100 to 300 micrometers in length, and the largest reported to be 750 micrometers. Compared to E. coli or a typical bacterium, which is only 1 micrometer, this is almost like 100 to 300 micrometers, cannot go up to 750 micrometer. So bacterium will be average of 2 micrometers, and which is only about 0.7 size of the Margareta nambiensis. This is something about a microorganism. But of course, there is another interesting question we all ask: what is the smallest organism in terms of, in case of microorganism which is present? And of course, there is one report, like several reports came after the report of Robert Fogg, who said that, like he's a sedimentary geologist who is also now considered to be father of nanobacteria. And he showed like there are organisms which are 0.05 to 0.2 micrometer in spherical structures and which are present. Actually, he reported them as a first mineralized nanobacteria or a DAF bacteria in the carbonate hot springs of Viterbo of Rome, Italy. So nanobacterium is by definition 1/1000th of a meter, 1/10th of a size of a bacterium living. But actually the question comes, like, will they be able to have necessary cell components like DNA, RNA, and plasmids? But of course, then there is another report which came from Australia by Professor Philippa Wins, and she collected some sandstones in Jurassic and Triassic sandstones from Australia in 1996. And then she was reluctant to call them as nanobacteria because she also asked the same question. Do they have DNA, RNA, and proteins, which can say something about their evolutionary history or the general presence? But of course, she stained those organisms with the TAPI and acridine orange and fusion staining. And then she was convinced that they are living organisms. And she reported her work in American Mineralogist in 1998. And that is a novel nano-organism from Australian sandstones. So we have some organisms which are of nano size. Of course, nanobacterial life has got interesting because this the simplest organism and will have necessary molecular pieces for life, may have RNA and ribosome and protein, and also which could also say more about the life in Mars in other places where we are looking for extraterrestrial life. Nanobacteria may mediate processes which are currently thought to be controlled by inorganic chemical reactions like precipitation of dolomite, oxidation of iron, and formation of clay minerals. Nanobacteria also may build an essential part of the larger organisms, play an important role in the disease of several organisms. In fact, nanobacteria is also considered to be responsible for kidney stone formation in the kidney. And there are some skepticism on this. There are some questions related to whether they are a fact or fiction, or maybe it is all self-replicating calcifying nanoparticles. The question remains, there are still people looking for answers. But to give you some more introduction, to introduce something about definitions which are available on nano-sized organisms. And there are organisms like dwarf cells, midget cells, and ultra small and ultra micro and ultra micro cells, where the miniaturization is actually due to the external factors, which could also result in the small size cell. Like if the environmental conditions are not so favorable, the cell size could also reduce. But there are some obligate ultra small, ultra micro, and nanoarchaea and nanoorganisms, where the cell volumes are about 0.1 micrometer. And then they will also have a genome sizes between 0.58 to 3.2 megabase per. And there are some organisms which are filterable microorganisms, which will be large enough microorganism to pass through the small pore micromembrane filters. So filtration will not going to help when in the filterable microorganisms. So if you look at the organisms, these organisms, a typical bacterium will have a surface area to volume ratio of 4.8, whereas ultra small or a nanoarchaeum will have a higher level of surface area to volume ratio. That is like in the case of Pelagibacter PQ, that is an ultra small bacterium, will have something around 22, and when you compare to the E. coli, which is only about 4.2, and a nanoarchaeum will have another ratio around 15. So this is going to give some advantage to the organisms. And also they have differences related to the number of proteins and then the amount of DNA which is available in these organisms. So organisms are really interesting because, when we talk about microorganism, most of the time we try to visualize microorganism in terms of a size and shape. So we always think anything which is around the region of 1 micrometer or plus and minus, say now about 300 micrometer to 750 micrometer, we accept them as microorganisms. But of course, there are also organisms which we could call them as nanoorganisms and nanos, and they are very smaller than 1 micrometer size. and they are also coming under microorganism. This is to give you some comparative pictures about related to the ultra small organisms, and then also filterable forms, which will be somewhere around this 250 nanometer to 350 nanometer, and something around like 300 nanometer to 500 nanometer you will be able to see. Whereas a bacterium will be almost like 1000 nanometer of size. And then we have several fossil remains, a nanoarchaeum, which is very, very small, which is even like 1 nanometer, 125 nanometer. So that means like we are talking about a group of organisms which are not to be defined by the micrometer sizes, beyond that. And also I showed you the size, the shape of these organisms as such a wide and diverse one. We wanted to put any particular definition for the shape. And now a size is also a difficult one to define for microorganisms. I'm just giving you a picture of Microbiome minutum, which is by Nakai. There's a paper which published in 2020, where he described about the ultra small and filterable microorganisms in the environment. And these ultra microbacteria, which are defined having a cell volume of less than 0.1 micrometer, cube micrometer, are often numerically dominant in the aqueous environment. So there are organisms in the aqueous environments which will be of ultra microbacteria size. And of course, like this particular RNT microbiome minutum, which is actually belonging to the filterable class of actinobacteria, Luna cluster, which was first reported by Hahn in 2003. So they may pass through 0.2 micrometer pore size to filter. So any filter which we have for to sterilize the liquids or any water and other sources, not going to be sufficient if you are using only 0.2 micrometer pore sized filter. You need to appreciate the life forms exist even at that small size, microbacteria. And also another interesting question which all of us will be concerned is, what is the largest living organism? It happens to be a fungus, which is known as Armillaria ustae, which actually attacks the barks of the trees and travels miles between trees. In 1988, they found out that a single specimen of this particular fungus can spread about 8.8 square kilometers, and this is estimated to be about 2,400 years old in Malheur National Forest in Oregon. And this is now aptly named as Mungus fungus. And this growth is considered the largest living organism on the earth. I'm not talking about the fossil organism. I'm talking about the living organism, which is about 2,400 years old, which is a fungus. Again, we call them as microorganism. So how do we see this microorganism? Initially, like nowadays, we learn a lot about microorganism, but in the past, although microscopes were available at the time of 1590, even at the time 1590, and the truly scientific use of this microscope was not made until 1661, when Marcelo Malbeghi discovered the capillaries in the dried lung of a frog. So this is the way microscope was, like just a toy which was brought into a scientific use. And there is another important publication by Robert Hooke, who wrote a book which is called Micrographia. And we are now going to be celebrating 355th anniversary of the publication, and he's also a contemporary of Sir Isaac Newton. He was a curator of the experiments in the Royal Society and professor of geometry at Crescent College. And he wrote a beautiful book called Micrographia, where he gave a description about the microscope which he used, and also about the small insects which he saw them under microscope, and beautifully gave the drawings. And this was published in 1665. You need to appreciate the amount of efforts they made in that year to come out with a book on small organisms. And of course, like we give the credit to the Dutch Antony van Leeuwenhoek as the first microbiologist in the world, because he was the one who actually used the microscope to describe the organisms, what we call as little animals, or what is now in the Latin word like animalcules. And then he also described the first bacteria and all those things he has drawn carefully, and he exchanged these drawings with the Royal Society in London. And now we actually consider him also as the father of microbiology. In fact, like the way he improved the microscope was something which is great, because on those days you could only make about 20 to 30 times bigger magnification of any object, and he improved it to 300 times magnification using his microscope. So he was able to describe all those small animals, including bacteria. So he was actually credited with the discovering bacteria, protists, nematodes, spermatozoa, and several other things. So we consider him as a father of microbiology. But right now we have several advances in those microscopes. So like some of you may be knowing about the compound microscope, epifluorescence microscope, confocal microscope, and so many microscopes. But also now we have atomic force microscope, which are used in a different level, so that we can also look at the surface of any biological object at the level of atom, or even a molecule level, to be on a safer side to say. And we have high-speed atomic force microscopy, optical imaging atomic force microscopy, and several improvements have come in terms of atomic force microscopy. I'm just giving you a picture so that you will understand the level of sophistication which is getting into microscopy now. There is one scanning and tunneling microscope which is in IBM, and which they say is the Nobel Prize winning tool, the first device that enables scientists to visualize the world all the way down to single atoms. So you are able to see even atoms, and which now with this microscope will weigh about 2 tons, operates at a temperature of negative 268 degrees Celsius, and magnifies the atomic surface over 100 million times. So you will be able to see many things, even to the level of atom, by using such scanning tunneling microscope. And then I'm just showing you a picture of an hydrogen atom, which was taken by a quantum microscopy. Is also possible. So microscopy is advancing in a greater way. So we can see a lot many things about the life and about life forms and their association using microscope. So the point which you all have to appreciate is what was once considered to be toy, a flea glass or flying glass by the Dutch people in 1590, that became a scientific tool. And then now we have several microscopes which are coming into that. And of course, I also wanted to show you like microscopy has improved to the nanoscopy, and we have a Nobel Prize was awarded in 2014 for several nanoscopes for the invention of nanoscopy. So along with that, like, once you are able to see these cells, and then we are able to appreciate there are several the underlying themes related to the life forms. One such thing is like cell theory. That was proposed by Robert Hooke and Antony van Leeuwenhoek. After their observations, they came out with a theory called cell theory, where all living things are made up of one or more cells, and the cells is a basic unit of life which all living activities occur. That was we are able to appreciate them. So all living cells come from previously existing cells. So the cell theory became very popular because of the hard work of Robert Brown and Matthias Jacob Schleiden, and also Schumann, and then Van Virchow. These are the people who contributed to the cell theory, and we accepted that like all of life as a whole. So we understand cells is the common unit of all life. And by similarly, we also have parallelly, there are several developments which showed that most of the cells have got some of the elements, and mostly like carbon, hydrogen, oxygen, phosphorus, and nitrogen and sulfur. And also we have several, biochemical compounds like amino acids, fatty acid, carbohydrate, and nucleotides. And this led to the one or another. Then, the important question was asked by Erwin Schrödinger. Like, he actually is a physicist, and he was giving several lectures in England. And in one of the lectures, he focused on an important question. How can the events in space and time, which take place within the spatial boundary of a living organism, be accounted for by physics and chemistry? So he also proposed the idea that a periodic crystal that contains genetic information in its configuration of covalent chemical bonds. So he could propose that there must be a chemical which is a configuration of a covalent chemical bond, and must be a periodic crystal, and which also having the genetic information. So that idea was actually stimulated several scientists, or created enthusiasm, and led to the discovery of the genetic molecule. And both Crick and Watson got inspired by his idea of a periodic crystal. In 1953, this double helix became so popular that it became a most recognizable icon in biology. And in fact, Crick's pencil sketch of a double DNA double helix you could see from his notebook. And that was where he showed about two anti-parallel strands, and that became so popular. Like in 1953, they published a paper that was just about one page, but then they claimed that Crick was so happy that he announced he found the secret of life. So everything, it's it's an important event in the biology, our understanding about the life forms. And in this modest paper, they also said, the structure has a novel feature which are of considerable biological interest. It's only a proposed structure. It is, they propose a structure, the proposal was accepted. In the sense, we all fascinated by that proposal. And then, of course, they also acknowledge that the knowledge and general nature of unpublished experimental results of results and ideas of Wilkins and Franklin, and they acknowledged. And they were given a Nobel Prize in 1962 along with Wilkins. In 1956, there was another important paper came from Clark, Cameron, Patterson, which is also very important to us because he is the one who actually gave the age of the Earth. So he was, he used radiometric technique, or rather he developed the uranium-lead dating method into lead-lead dating method. And using the lead isotopic data from Canyon Diablo meteoroid, he could calculate the age of the Earth as 4.5 billion years, 4.55 billion years. And that's, even now considered to be the accurate figure, and it is not changed since 1956. So we have, this 4.5 years ago, the life has formed. And of course, we had the first organisms coming up in 3.5 billion years ago. And there are several things which you could understand from the evolution of life. And this paper, which appeared in 1956 in Geochemica, Cosmica, and where he showed that the accurate method by which he could also, see the age of the Earth as 4.5 billion years ago. And that is an important for us to appreciate the evolution of life itself. And then by then, we could also understand that the stunning variety of life systems belies a striking similarity. There is a lot of similarity, although there are different forms of life. The common use of DNA and the genetic code by all organisms underlies one of the most powerful discoveries in the past century. And all organisms are built from similar molecular components, although there are minor variations. That uniformity reveals all the organisms on Earth. To give you information about the age of the Earth, or how the planet Earth was formed, or how many years have passed So this is something which we have to appreciate along with the appearance of a common ancestor. And of course, the microorganism also contributed immensely to the discovery of several secrets of life. And we have, Jacob and Menard, who proposed the existence of mRNA, also gave the theory of, the operon theory. And that, parallelly, we could also understand about the genetic information being, passed down from one generation to the another. And there are mutational events which could also make changes in the phenotype of the organism which received the genetic material. And all this led to the important, it's what was actually considered, it's a difficult question. Can we compare apple to orange, which is not possible by a human being? But again, it's possible if you look at them at the genetic level. That made the Jacques Menard to say what is true for E. coli is also true for the elephant. So the Mendelian modern synthesis or natural selection of the organisms which is acting on the mutations generate new adaptation and new species. That means, we could also have the natural selection is an important driving force for all kind of lives which is getting evolved on the earth. And at the same time, in 1960s, there was a theory proposed by Lynn Margulis, who showed that the major organizational event in the history of life could also involve the merging of two or more lineages through symbiosis. So by sheer observation of the cells and then of the chloroplast in plant cells, and then also like about the mitochondria in the eukaryotic organism, she could say that like there is a merging of two lineages through symbiosis. So that is an important concept, because the origin of mitosing cells, where she showed like using cytological, biochemical, and paleontological evidence, she proposed the theory called how eukaryotic cells are formed. So the point is like we have organisms which have developed by engulfing the aerobic heterotrophic prokaryote, and then they will act like a mitochondrion. Then we have a heterotrophic eukaryote. When they engulf mitochondria plus a photosynthetic prokaryote, they will also give out to the ancestral photosynthetic eukaryote, and which is we call them as, now we understand that these is possible in case of plants and other organisms. And this, the theory was actually supported in the sense, molecular evidence was given by Schwartz and they have in 1978, where they clearly showed the origin of prokaryotes and eukaryotes and mitochondria and chloroplast could also be appreciated by the sequence similarities and their comparisons. So, and then after accepting that like the macromolecules like DNA, RNA, and proteins are common in all the living organisms, people started looking for how to identify and like use those information for understanding the relationship among those organisms rather than going by size and shape. So size and shape is a meaningless one. So we can only understand about the life similarities or their, how they are interrelated to each other by looking at the molecular chronometers. So Carl Lewis was a professor in University of Illinois, a biophysicist, and he used those sequence information of DNA, RNA, and protein, and in fact, like he used only RNA, and he could also, and then say like how organisms have evolved, and he proposed the three-domain concept, and this is very important in the sense ribosomal RNA is universally distributed among all the group of organisms, and they are functionally similar in between organisms, and the mutational events are very slow, and the sequences of RNA could be aligned and it can also be compared. And that, then the phylogeny or the tree of life concept was proposed, which was proposed by Carl Lewis, and that was actually made used by Norman Pace in the environmentals, in different environments, and he showed that rather like distribution of several bacteria and archaea in the environment, and also said every species alive today has been perfected for its niche through millions of years of evolution. So evolution is the radiation, not a ladder. So it is all through, millions and millions of years of evolution has led to the appearance of a particular species, which is perfected in a particular niche. That idea we need to appreciate. And by then, we also realize the importance of molecules. And as I said to you, sequencing has become very important. In fact, the man who sequenced protein also took interest in the sequencing of nucleic acids. But initially, he thought DNA as an inert substance. And he said that DNA contained all the information for making a complete organism would have been thought of as a science fiction. So he didn't believe in DNA. But later on, he joined the Laboratory of Molecular Biology along with Crick, and he himself said, with the people like Francis Crick around, it was difficult to ignore nucleic acids and failed to realize the importance of sequencing them. So once he joined the Laboratory of Molecular Biology in Amherst, and he started putting all his energy into sequencing RNA. And in fact, he worked out a simple way to synthesize radioactive RNA, and 32P inorganic phosphate was used by growing or yeast and E. coli, and then he was able to isolate them. This is a page from his record, where a laboratory notebook where he showed how to prepare I'll just check this. Where he showed and then he also announced the successful sequencing of another short RNA, 5S ribosomal RNA from E. coli, and that was started. Then he started using to determine the sequence of a single-stranded circular DNA from 5X174 bacteriophages. All those interesting development you could read from his paper, sequences, which appeared in Annual Review of Biochemistry. So that made, and then he proposed the dideoxy sequencing procedure. Learned from many of the textbooks on that, and he, in fact, Sanger was given a Nobel Prize 2 times, and he was very humble enough to say it is much more difficult to get the first prize than to get the second one, because if you have already got a prize, then you get all facilities for work, you can get collaborators, and everything is much easier. In 1980, he got a second prize for the sequencing efforts he made for DNA, nucleic acids. Of course, there are several developments now. So next generation sequencing, we have Illumina, HiSeq, and then ABI, Sanger, and so many things are available. So you can understand, people understood sequencing methods and equipment, which is making sequencing cheaper and cheaper now. So we all understand, we could all, all these efforts are actually in a way to understand the secrets of life. So everybody wants to sequence a nucleic acid or DNA, RNA, and to understand what we have and how we could understand about the life. And to make a point, this led to the revolution of evolutionary biology also. So we slowly moved from microscopy to molecular sequencing. So all our depends on microscopy is slowly changing now. So by looking at the sequence of DNA, we are able to appreciate the life and the presence of life itself. Also you can see the relationship with the phylogenetic relationship with all other organisms. So what was once considered the genome is about 95.3 million, but now you can get it by, in 2017, you can get it for $1,121. You can get the whole genome sequence. So that is a level of improvements which have come up in the case of sequencing. That also made things easy for microbiologists. That's a point you should understand. Once we could follow the molecular chronometers, and also 16S ribosomal sequencing, RNA sequencing, and another sequencing made things easy for us to compare, to appreciate the different forms of life. So I'm just going through a couple of slides where we appreciate the form of different forms of life in the earth. So one is like, although we know about methane as a gas, chemistry of methane was reported by Alexandra Volta in 1776. And also we understand like methane gases are emitted from rice fields in 1913 by Harrison and Dyer from a publication which from Indian Agricultural Research Institute, where they showed about the gases of rice soil, their composition in relation to the crop in 1930. And we didn't have several methods to study the organisms which are responsible for methane formation in rice soils or soils. So we relied heavily on microscope for a long time, and also on the culturable methods like vertebrate culturing of organisms. And of course, in 73, Hungate came out of the roll tube method for cultivation of strict anaerobes. And then we are able to appreciate the different forms of organisms which are methanogens, because since we live in an aerobic world, we need 20% oxygen to survive, and we do all of our activities under oxygen atmosphere. We never appreciate the life forms which exist in the anaerobic organisms or the anaerobic environments. So I'm just giving you how we could appreciate about the presence of anaerobic organisms, especially methanogens. And methanogens was actually first reported by Marjorie Stephenson in 1933, where she could show that bacterial formation of methane by the reduction of one carbon compounds by molecular hydrogen. And in fact, like it took almost, say about 55 years to isolate one such bacterium from rice paddies by Rajagopal and Lassie Daniels in USA, in Texas, where they isolated methanogen from rice fields. Now we have several reports to show that like methanogens are present, and even from Chinese rice fields by Methanacella condradi, and which is published in 2012 by Plus One. So we have several organisms. And I had an opportunity to look at my archaeal community in terms of TRFLP, and I could see that like there are several organisms which, you know, in 2000, and we had only two what you call kingdom in those days. We had only Euryarchaeota and Cranoarchaeota. And we also had clusters like rice cluster 1, 2, 3, 4, 5, and 6, and which are unculturable at that time. And we could say that like they all belong to the archaea, and some of them are capable of producing methane. And these key methanogens are present in different soils, which we isolated from rice fields in China, Philippines, and also from Cameroon, South Africa, and Switzerland, and several other places. That means they are distributed globally, and these organisms are, you know, we are able to isolate them. So see, there are several interesting questions even now. The point is, and by now we have some understanding about methanogens. Methanogens, they evolved around 3.5 billion years ago. And the major question is, how do they survive after so many years of rice cultivation in soils? Because every time we expose them oxygen, and they are still surviving as an anaerobic organism in these soils. And what are the factors that that affect the activities, and how do they differ in the roles in carbon cycling in different rice growing regions? It's also an important question which we need to understand. So, and once now we know that like we use the molecular markers, or what you call molecular chronometers, once we are able to sequence the 16S ribosomal RNA. And we now understand like bacteria, there are about 92 phyla, and archaea, there are about 26 archaeal phyla. And then this view of tree of life is getting changed as we know new information is coming from different environments and niches. So what was once known as gram-positive, gram-negative bacteria are rod cells, and then we have different shapes like cocci, and all those things are now slowly changing. So we appreciate the forms of microorganism in terms of their presence, by 16S ribosomal RNA sequence or the metagenomic sequences which we get from different environments. So why are we concerned about the microbial diversities? Like now, once we know like what are the different diverse forms of microorganism, we will be able to understand the novel metabolisms, as I said to you. So anaerobic forms of organisms was, although we know in the laboratory, or maybe in some kind of lactic acid fermentation and other, alcohol fermentation, by Louis Pasteur and others, but like their presence in the environment was not appreciated unless we have defined a new refined tools to see the microorganisms. And the unknown microorganisms are also largest potential reservoir of useful genes for medicine, agriculture, and biotechnology. So the knowledge of microbial diversity is essential to understand the evolution of life forms. So my idea is to tell you that like all these, how these life forms are connected to one another. So they are not island, they don't survive as a single entity. So to give you an idea about archaea, so let me not take you to the bacterial phyla, which is 92 around now. It's a little complex to understand about the relationship, but at least I'll tell you the kind of developments which happen in archaea, so that you will be able to appreciate like how our understanding is growing by decades or every year, how we get a new information so that we are able to appreciate them. So as I told you earlier, in 1990 to 2000, we had only two kingdoms. Like those days, we used the term like Euryarchaeota and Crenoarchaeota, and slowly they added the tag where they Thamarchaeota, Agararchaeota, Crenoarchaeota, Cororarchaeota, and Nanarchaeota in 2002 to 2011. But 2011 to 2017, now we have too many other phyla like Euryarchaeota, tag. This is what we call them as Protearchaeota and Asgard. This is something like Lokiarchaeota, Thorarchaeota, Hemidolarchaeota, and also DPAN, like what we call as a Diaphoretetis and Altiarchaea and other groups about here. So the point is, our knowledge about the methanogens or archaea is also like methanogenic archaea as well as archaea is slowly improving because we're able to get hold of related to diversity in different environmental niches. So what is more important is like their key metabolic capabilities also we have to appreciate, because some of this Euryarchaeota and tag, we have methane and beauty metabolism capacity. So they are methanogenic in style, and some of this archaea is also like aerobic lifestyle is also seen in tag and then also in Asgard. And some, they have the capacity to fix carbon using Wood-Jungdahl pathway and have a reverse Gyrus pathway. So more we understand about the diverse forms of these organisms, we will be able to appreciate about their metabolic capabilities and chemical reaction they are capable of doing it. So I take you to the concept of cellular life origin. So we have several proposals, but we all now come to an understanding that the last universal common ancestor is an anaerobic organism, which is a carbon dioxide fixing, hydrogen dependent with the Wood-Jungdahl pathway, and it is also nitrogen fixing and thermophilic organism. So this was proposed by a group from Germany. I think the paper came from Germany, and they were able to show that like the common ancestor is having just about 355 genes compared to E. coli cell, which is having about 4,200 odd genes, and this is having only about 350 genes. So the point is, we have organisms which are capable of making a living with just about 355 genes. So that we consider as the last universal common ancestor, and which has evolved to give several other forms of life. So I just want you to appreciate like how microorganisms get their energy. So microorganisms are very special in several ways because they are able to get their energy by making use of the redox gradients. Redox gradients are nothing but the ability to transfer electrons between oxidant and reactants, and they give the metabolic energy for this organism to survive. So they are able to use several what you call redox couple. So they have a negative and positive couple of several compounds. So once we know that the Earth is having several such electron or more redox couples, which is with oxidized and reduced compounds, organism can make use of these compounds by transferring the electrons from one to another, and they may get the energy and ATP, they are synthesized and they are able to make a living. So they don't have to bother about only photosynthesis, but they do live by using several such chemical processes where they just transfer the electrons and they use the redox couple and then make a living. So we have now interesting several, like some of us must be knowing about the type of organism, microorganism, which is like photoautotrophs, chemoautotrophs, chemoheterotrophs, photoheterotrophs, and chemoheterotrophs. So by the way they are able to fix carbon, and also by using different sources of energy, so we were able to classify them into several groups. And so that means like the great diversity of electron donors and electron acceptors in the earth in any environmental sample means microorganism also need to be a good chemist, and they are. So you go to any environment, you will be able to find microorganisms. So diversity of microorganism, well, we appreciate very well. Soil is one important medium or important home to several microorganisms. So if you look at them, like, we have, 1 teaspoon of healthy soil will have about 100 million to 1 billion individual bacteria. That's what you can understand. But if you don't have any organism in the soil, we can only call the soil as sterile soil. We don't call them as fertile soil. So we have importance of microorganisms in the living organism in the soil is understood. So we have soil bacteria, soil fauna, and other organisms which make the soil as a fertile medium for plant growth. We also determine the soil health using the biological principles or biological parameters, and we provide the cards to the farmers to appreciate the health importance of soil health and their contribution to soil fertility. But this is not just the one aspect of soil diversity, but the another one is these microorganisms present in the soil, we all have to appreciate that like Earth is home to almost 1 trillion microbial species. This is what the proposal which came in VNA is by Lassie. And they use the scaling laws to show that we have about 10 to the power 12 microbial species. It's not just few species, but so many number of microorganisms are present on the Earth. What do? And if you look at the number of cells, in the Earth, we have about 4 to 6 in 10 to the power 30 cells. So they are important reservoir of carbon in terms of petagrams. 1 petagram is 10 to the power 15 grams. So we have something around 350 to 550 petagrams of carbon. And they almost, what is present in the plant carbon is also present in the microorganisms. So microorganisms in the soil or in the earth, they have equivalent number of equivalent amounts of carbon, and also they have nitrogen and phosphorus, and which are having about interesting cells turnover of 1.7 into 10 to the power 30 cells per year, and with the 60 to 25 days, they have a turnover. So this organism has got important role they play in the food web. I'm not talking about the food chain. There is no one after another, but here it is web. Everything is interconnected. So microorganisms play a very important role in the food web of, food web in the soils. So we have now the importance of these organisms are understood. Globally, there are several efforts where they wanted to understand about the microbiome. So there are several projects where, something like Earth Microbiome Project, and then we have Terrestrial project where soil metagenome is, sequencing. It's all based on the basic principle that DNA or RNA is the genetic material, and the sequence information will also give information about the diverse forms of organisms which are present in the different environments. Okay, so microbiome is now, it's a concept which is actually used not just the organism, but also about the genome, genetic information which is available in different organisms. Since I told you like cells and sizes, cell size and shapes do not matter much for the microorganisms, we see in terms of the genetic capabilities, of physiological, functional capabilities, and then in the microbiome concept was proposed. So we have now, when you consider the organism in terms not only the diversity, but also about the genetic potential, then we call them as microbiome. So anything which is in a particular environment, where the number of organisms as well as their genetic potential is considered together, and we call them as a microbiome. So it involves microbiota of bacteria, archaea, fungi, protists, and algae. And also they have the microbial structural elements like proteins, lipids, polysaccharides, and nucleic acids. And their, then internal, external structural elements like mobile genetic elements and their interactions, everything is put together, is considered, it is considered. So the point is like, we are now interconnected. We are not just one organism. Like I said to you in the beginning, superorganism. So every environment, every organism, every living organism is connected with other organism, if not to any other higher other organism of the same species, but at least to microorganisms. That is a point we all have to appreciate. So no organism is capable of living on its own. Maybe few microorganism can live. I'm here to say like they are also able to live independently, because several organisms, they live together as a biofilm, or they live together as a community. But like planktonic, single organism cannot make a living for a long. They need to have a support of other community organisms in order to divide, like what we call division of labor, and also to utilize one metabolite by another. So they need all kind of interconnection. What we need to appreciate. So no organism is an island. It's all interconnected. So that we could understand by the microbiome. So now what we are now, essentially we are trying to understand is who is there. What are the microbiota which are present in a particular environment and organism? What can they do and what are they doing? So we have several methodology or strategies. So as I told you in the beginning, microscopy played a very good role, significant role in understanding the life. Understanding about the microbial life and other life. Also about culturomics in case of microorganism. So we have metagenomics or metabarcoding in case of DNA, and metatranscriptomics, metaproteomics strategies which are being used to understand about protein, and also metabolites using by metabolomic studies, and metabolites can be understood. And then the interaction could also be different. There could be cooperation and competition among the organism. So there will be a positive interaction or negative interaction, which makes all the differences in the formation of biofilm formation or the quarant sensing, where another organism can get together by sensing the molecules, and then they can form a kind of they can come together. This paper on microbiome definition revisited is published in the journal Microbiome in 2020, which you can have an access. So the point is, right now we are all like six blind men and the elephant situation, where each blind man will be describing the elephant in his own words. In a similar way, we are also describing the microbiome of any organism or any environment. So we are trying to understand the organisms, microorganisms associated with each of this environment. But of course, I'm hopeful that one day we are able to put all these things together and understand about the microbiome of each environment and organism. So I'll also give you a few examples where our interests are now focused on the role of microbiome in plant. So we were focusing the role of microorganism in nutrient cycling in the rhizosphere. So we know there are few symbiotic organisms, like they are like not only in bacteria, in the plants of legumes or pulses, and they are able to fix nitrogen. And we also know that there are few microorganisms like mycorrhizal fungi, and they form mycorrhiza in the roots. And there are several organisms which are commensal, and they form an associative symbiotic relationship with the roots, and they play an important role in the nutrient cycling and protecting from the plant pathogens, and also give you some kind of an immunity, may be able to give a tolerance to several stress conditions. And there are philosphere, surface. You have microorganisms present on the surface. They actually protect UV rays, the transpirational loss of water from the philosphere or the leaf surfaces. And endosphere, where the plants, inside the plants, we have microorganisms. So describing all these organisms is very difficult. So what we do is that we use a reductionist approach. We take each part independently or selectively, and then study about individually which component is having what type of microbiota and what are they doing. It's a real typical job we are all now facing. So microbiome challenges are now too many, and since if you look at the microbiome itself, if you compare the plants of Arabidopsis, maize and rice, you will understand that the abundance of different bacteria could see they are actually at different levels. So Arabidopsis has got a different level of proteobacteria, whereas rice has got a different level, and actinobacteria well of similar level. Maybe acidobacteria, they're completely a different. Bacteroides, a different level. And then we have cyanobacteria more in the case of maize, and whereas less in the case of rice. And we have Microbia, Nitrospira, and chlorofluxi. All these things are different. So that means there is a kind of a preference by each plant species to have a certain level of abundance in the rhizosphere that they contribute to the nutrient cycling, tolerance, stress tolerance, and also to the plant immunity. And now we call them as second genome. These associated microorganisms with the plants are all called as microbiomes, and they actually are called second genome. The host genome is largely the genetic material which is present in the plant species. In addition to that, the genes which are available in those organisms associated with the plants, microbiota, or we call them as second genome. So that's important, and there are several factors which will contribute to the association between the microorganism and the plant species, which will be dependent on host genotype, developmental stages, plant health, plant fitness, which is on the above ground. And in the below ground, plants actually give out different amounts and different nature of root exudation. They have a chemotactic properties. They recognize the bacteria. They allow the bacteria to colonize and then make biofilm. And by those strategies, they form an association with the plants. So this is what we all have to be, we have to make we have to get a clear understanding of the role and by the microorganisms. So in the past, we were actually using individual cultures and then looking at the association using a plant binary assay, plant and microorganism. But now we have the tools like nucleic acid sequencing based on 16S ribosomal RNA, and could understand about the association of microorganism in the different components of the plant. And this microbiome change of the plants. If you alter the microbiome, and you will have a different level of induction of systematic resistance, induced systematic resistance, ISR, and also the levels of root to shoot and shoot to root signals, and the phytohormones which are produced by plants also could be altered by using different composition of microorganisms. And you can also make changes in the antimicrobials produced in the rhizosphere or in the philosphere by changing the composition of microorganism or the microbial communities associated with the plants. And of course, biofilm formation could also be changed by these associations. So I'm giving you one example where you can understand that the microbiomes could also make large changes in the flowering time. You can manipulate or modulate the flowering time by changing the microorganism associated in the rhizosphere. So since microbiome, rhizosphere microbiome, or the organisms which are now preferred by the plants, they produce phenols, amino acids, and IA, indole acetic acid, and then they are involved in nitrogen cycle. And they make the changes in the plant, the phytohormone production, and then also root shoot signals or shoot to root signals, and then they change the way the plants can flower. And this interesting paper was reported by, in the ISME journal in 2015. Also in another paper in the Microbiome by Lu in 2018. So that showed that microorganism has got a greater influence on the plant flowering and several phenotypes. And well, I show a couple of examples about human microbiome. The website of BBC Earth, and then look for what is your worth. where you can enter your details and come to know about the number of organisms you possess. And we have about 100 trillion organisms. And then, of course, they are all important because they are able to regulate our immune system, help to coagulate our blood by producing vitamins like vitamin K, B, B12, riboflavin, and thiamine. And well, that's, I also wanted to show you about the connection with the brain. And the microorganism which are present in the gut, they have an interesting connection with the brain. And if you look at the human brain, they are only almost like when you compare with the elephants, if elephant has got only, 0.1% of its biomass as a brain, whereas chimpanzee has got 0.8, whereas human had got to almost 2% of his body weight or body biomass as brain. And if you look at the brain, you have several what you call folds in the human brain, and what we call them as what you call the cerebellum. This you could also see in a dolphin and human. So those two are considered to be closely like very intelligent animals rather. And the microorganism which present in the human gut, they play several roles, and and then they can also change the way brain behave. And in terms of they give out peripheral serotonin, modify the immune system, and some of the bacterial molecules can send you the signals, and then they can modify the way the brain behave. So don't underestimate the microbiome of your gut. To give you an example, if you have a very unpleasant food item, and you will have a very bad mood, and the bad mood is normally because generally it's because of the changes you have made in your gut microbiome. If you irritate your gut microbiome, you will have irritable behavior. So that you could easily relate. And of course, there's another example, like if you are not keeping well, and we go to a doctor, we have antibiotics to get rid of the bad bacteria, or a skin infection or some other organ infection. And if they change the microbiome in the gut, also you will have problem in the behavior. So that will put you into kind of an unpleasant situation. All those things are all related to the changes you make in the gut. So microbiome has got an important role in the gut brain axis is now clearly understood and appreciated. There are a couple of slides where I will show you the changes in the lung microbiome could also result in the acute respiratory distress syndrome. May make you difficult to respire, difficult to make a living. So one such thing is like right now we are all worried about COVID virus, SARS-CoV-2, and if it gets into lungs, we know they make a lot of changes. And of course, they probably get rid of several microorganisms which are present in the lungs, making very unpleasant or rather very difficult to breathe because the changes they make in the lungs. And if the gut microbiome, due to some sepsis and other reasons, like if they get into the lungs, you have a pulmonary dysbiosis. And maybe if you have an oral tooth decay and create a different kind of a microbiome in your mouth, can get into your lungs, you're going to have several problems with the microbiome associated with the lung. And of course, that will give you a disease called acute respiratory distress syndrome because of the changes you make in the microbiome. And I'm just showing you a few pictures where the scanning electron micrograph of skin microbiome. This is the microorganisms which are closely present in the sweat gland, and these are colored. You may not be able to see them in the same way, but they are colored for the purpose of understanding about the organisms which are present in it. And this is on the tooth. When we have a dental plaque, the pink plaque which is present in the tooth, and you just see the kind of image we are able to get with the scanning electron microscope. And when you see these pictures with those of Anton van Leeuwenhoek in 1674, where he showed you the organism animalcules from his tooth, and see the way we are able to see them now. Was large improvements in seeing those organisms present in the tooth. And this is a kind of advances. These kind of advances will lead to make several, understanding about the association or connection of microorganism with all other organisms. So when I talk about microbiome and genome, I give you an idea, a rough idea that if you look at the human body, we have something like 23,000 genes, but the organism which are associated with you will give you something around like 30 lakh organism genes which is being, like they actually help you to face several what you call stress, are able to grow. So the point which now they are trying to, even now they are come to an understanding that although human genome sequences or information could be similar between two individuals, if the microbiome of those individuals are too different, you will see a large differences. You will be able to see large differences in the way they gain weight, they behave, and the way they move. Everything is going to change because of the microorganisms which are associated with the human body. So, and I am not going to limit only to human beings and plant system. I want to even show you like the importance of microorganisms, and not understood only by us, they are also understood by ants. And I'm just showing you a picture where ants are able to cultivate and then make use of those plant leaves and microorganisms for their own living. So the evolution of agriculture is not related only to by human beings. Agriculture was practiced by ants for several millennia, if not by millennia. So agriculture, the concept of agriculture is not something new, not only to human beings, but also to the ants. So these ants are able to select the microorganism, grow the fungal garden, and feed those fungal garden to their own younger ones. So they are able to withstand or rather lab antibiotics and then get protection from diseases or pathogens. It's also known to the ants, which now we are able to appreciate. So microbiome is not something only to, which is meaningful to us, to other organisms. I just give you another example where butterfly microbiome was studied. They are able to say like there were several organisms related to gamma, beta, proteome, alpha proteome bacteria, which are actinobacteria, which are associated with the butterfly. So we have the whole insect development, caterpillar to the butterfly. You are able to see their association with the microorganism. So when in a recent study, legacy of a butterflies parental microbiome offering of performance, published in 2020, recently came out an article where they changed the butterfly caterpillar with the antibiotics, and then they moved to the other sources either without antibiotics, and they saw the changes in the microbiome, and could also see several changes related to the abundance of or phyla related to Acidobacteriaceae, and then Lactococcus, Serraceae, Enterococcus, and Enterobacteriaceae, and Escherichia, and other organisms. So you have to appreciate the organisms are associated even with the butterfly. Maybe one day we will come to know like how do they, help these organisms to not only for nutrition, but also like in sensing in the different flowers and others, so that they will be able to fly out and then feed on them and grow. Well, I give you another example where insect microbiome is being now seriously considered for applications. Like we have mosquitoes and malaria is very, it's a biggest threat to the human being. If you don't consider about the recent COVID, I will say malaria is the deadliest form of, rather, mosquitoes are the deadliest animal, and they cause, malaria is actually causing more human death than any other, any other accidents or any other diseases. Malaria is such a notorious disease. And then this human malaria is transmitted only by females of the genus Anopheles. And there are 430 Anopheles species. Only 30 to 40 can transmit the malarial, or rather they can serve as a vector. So this, and if you look at them, like this is the mosquito, like the female mosquitoes can take the blood meals to carry out the egg production. And such a blood meals are linked between human and the mosquito host in the parasitic life cycle. And interestingly, there are populations where of mosquitoes, where the native microbiome can, prevent the vertical transmission of Wolbachia in the mosquitoes. And also there are naturally group of microorganism, which is now recently considered to be a promising strategy to prevent the arboviral transmission. And then they wanted to control the mosquitoes by making those all the females of mosquitoes with the Wolbachia, so that they become they only become all females or all males. Like that, you can make changes and control the mosquito effectively. So so microbiome is also being applied in the sense can control for the mosquitoes in the future. And I just wanted to show you a couple of other ideas where I want to share with you, so that you will appreciate how the transfer of genomic information is now, is possible. When I was a student, and my professor asked me, is it possible that you can find the transfer of genetic material between prokaryotic and eukaryote? I said, yes, after all, it is a DNA. But then he said, no, there are several barriers. Also, I learned at the time there are several barriers. And those barriers are related to the availability of DNA, uptake of heterologous DNA, and in the instability in the new host, and there are restriction enzymes which will cut and chop the DNA which is coming from other sources. So it's not possible. And the stabilization issues related to the heterologous DNA. And also there are possibly expression of heterologous DNA and other things. And there are several other barriers related to splicing, RNA editing, and then GC and codon usage, incompatibility, all those factors are available. Even then, there are some gene swap which is going on. One such example is the Wolbachia gene transfer between Drosophila and Wolbachia. So they are able to get a genome, from the insect, and the gene gets transferred into the, to the Drosophila. So we have several mechanisms of DNA integration. So genes are now getting double insertion, and then transport-mediated insertion is possible. So non-homologous repair is also one of the ways by which the DNA from the bacterium can go into the organism. So interconnections are not just at the organism level. They could also occur at the gene level. It's possible. So another interesting finding was how many microbial genes are in the human genome. The question is, we all know the gut is having several organisms, microorganisms. I also said skin is having it, and all other organs. Maybe there are a couple of reports which are showing that even brain is having microorganisms, microbiome of brain. Well, it's not fully understood, but of course there will be a day. We will come to know how much of microorganisms which are present in the brain. But of course, there is a barrier which is actually, which will not allow the microorganism, the blood, barrier. Blood from the blood, it cannot get into the head. That's what we now, understand. But maybe later on we will know about the changes which is happening in the brain also due to microorganisms as such. But okay, coming back to here, if you look at the human genome, there are about 40 genes which are actually shared between humans and bacteria. Came up in 2001. So several genes are actually shared between the bacteria and the human, or the interrelationship, or the association which are making impact or the influences on the human physiology or human behavior or human phenotypes, but also the genes exchanges do happen. They are very important. Of course, there are a couple of other incidences where this kind of exchanges are not pleasant, in the sense some of those exchanges can also lead to cancer, because if you go into the lateral gene transfer of the bacterial DNA, then cells integrated with that DNA can become cancerous. They can form tumors, and that's what you are able to see in the cases of acute myeloid leukemia, and where Acinetobacter species can integrate with the human genome and cause a tumor, and where some in Pseudomonas in stomach can cause adenocarcinomas transample. So maybe that association is not present in case of some bacterial exchange of genetic material. So well, the point we all have to appreciate now is life has evolved from the last common universal ancestor, universal. We have several organisms now appeared, evolved, then become extinct. Then we still continue to make our own evolution and going on. But we also should appreciate that several of these evolutionary processes are all because of the appearance of life forms in somewhere in in the case of microorganisms, which happened in the Cambrian era. Well, now if you look at any of this biosphere, we understand that the level of hierarchy of the hierarchy level of interactions are like from atom to molecule to organelle, cell, organ, and organization, population, community, ecosystem, and biosphere. So right now from here, from after molecule, it is all like microorganisms they play at every level. So we should appreciate about the microbiome in all the environments with all the organisms. So I'm actually sharing you about like what we understand about microbiomes is what we know about the knowledge and information, and there are known knowns. We understand those things that we know, and there are known unknowns. We know that there are few unknowns are there. And there are also unknowns. We don't know what they are and we don't know what they are. So this is an interesting thing in the sense, a lot to learn from these forms of organisms and microbial association. I just wanted to thank all my senior colleagues who inspired me to take a part in this microbiology, and also to my students and my colleagues who helped me who help and then travel along with me in our understanding about the microbiomes. Well, I thank you for your attention, and I hope you will also get inspired to know more about the role of microorganisms or microbiomes in each environment. Thank you very much.
Thank you, sir, for a very informative lecture. Now the session is open for questions. Before going for the question answer session, I want to make one announcement for the participants. The certificate generation link will be posted in the chat box. Participants, please fill the form and get the link to generate the form, please. Sir, now we have a question.
Okay.
Sir, microbiome is unique to different environment. Is there any microbiome pertain to climate? This is the question from Dr. Jnana Chitra.
Microbiome related to what?
Climate.
The climate is a word term which is something difficult to associate with the microbiome, because climate is events which happen over a period of time, weather events. Okay, and if you look at the way Earth has evolved, there are unique microbiomes at every stages of the Earth evolution. So in the beginning of the Earth, Archean, it is all anaerobic organism in the beginning. Okay, only when the great oxidation event happened, then cyanobacteria started proliferating and they are able to give out oxygen. Then oxygen become it's almost came up to 20% in the atmosphere. Then we have all aerobic organisms. We are also benefited by the cyanobacterial oxygen evolution. So that's why aerobically we are able to live, and anaerobically we can't make a living. So microorganisms, in the Archean periods are totally different. Microorganisms which are present in the after the great oxidation event in the evolution is totally different. This is one aspect about the Earth evolution. But also there are microorganisms which are now considered to be involved in the cloud formation. So how rainfall is happening? Again, the organisms which are present in the clouds are making some kind of changes in the clouds and then rainfall events. So I'm not sure about like how climate is going to be totally changed, but all those what you call greenhouse gases like carbon dioxide, methane, nitrous oxide, and well, these three group of organisms and others, all actually being emitted by the microbial activities. Carbon dioxide is aerobically respiring bacterium, and also by anaerobic organism can give out carbon dioxide. And anaerobic organism give methanogen. Methane is coming out from anaerobic organism. Nitrous oxide emission. It's again by denitrifying organism and other nitrifying bacteria. So they play an important role in changing the atmospheric concentration of greenhouse gases. So they can alter the climate. So linkages are too many. It is not just the microbiome with the climate, but climate itself is actually being influenced by the activities of microorganisms. Hope she can understand, I mean, she can understand, or he can understand what I'm trying to explain.
Okay, sir. So the next question, is there any application for human welfare reported this of my nanobiomes?
Nanobiomes, I'm not sure. Nanobiome, there is no application so far, but microbiome, yes. If you see in your TV, sometimes you will see an advertisement called Yakult, where people will talk about bifidobacterium and lactobacilli as a probiotic concentration, that can improve your gut health. And then you will have a pleasant stomach, or you will have a pleasant feeling. And those probiotic organisms are microbial products only. So microbiome changes could also be made by using probiotic. But nanobiomes, nanobes, nanobacteria, still now no. But of course, people speculate that like they are responsible for stone formation in the kidney.
Okay, sir. Sir, kindly throw light on latest trend in research on biological nitrogen fixation.
Biological nitrogen fixation is an interesting one, but unfortunately, we contradict rather, we clashed with the biological process by chemical synthesis of fertilizers. Once ammonia is produced, in the sense once we learn how to make ammonia, paper process, and we are now producing huge amounts of fertilizers, ammonia, urea, and all other fertilizers. And when we apply these fertilizers to the agricultural fields, and we are actually driving out the biological nitrogen fixing organism. In the last 60 years of fertilizer use, we are now losing several nitrogen fixing bacteria from the soil and other environment, because pollution of nitrogen, nitrate, and ammonia is actually detrimental to the microorganisms. So we may face a lot of problem with the biological nitrogen fixation. And of course, it is a challenge. And this, in a way, we have to promote biological nitrogen fixation, because we don't want to contaminate or pollute the earth with several nitrate pollutants or ammonia, nitrous oxide pollutants in the atmosphere.
Thank you, sir. So the next question is from one participant. Yesterday, they found a bacteria present in our body, large intestine, which will cure heart disease. What do you think about it, sir?
There may be a role. What is the bacterium? I don't know, but there are several bacteria like gut microbiome, bifidobacterium, and Provetella, and other group of organisms which are playing positive role in the heart and gut relationship, because well, I expect that there will be some more understanding about the role of microbiome, gut microbiome in the heart health, and as well as brain and other organism health, because everything is interconnected, because all the nutrients which we get, all those are from the gut only. It's like they play a very major role in determining the health of organisms. Yes.
Sir, what are the parameters for biological soil health card?
If you visit the soil health card website in the Government of India, Ministry of Agriculture and Farmers Welfare, you will come to know the parameters which are being used. So biologically, we have several parameters which are listed, which are related to microbial activities also, like substrate respiration, induced respiration, and dehydrogenase and other enzyme activities. But it has to be improved upon using the microbial diversity information. That needs to be done. But right now, several of biological, parameters related to soil functions like dehydrogenase, alkaline phosphatase, acid phosphatase, and then like denitrification activity, and all those things are being considered for biological health.
Sir, can microbes play the role of keystone species in an ecosystem?
And definitely, they will. If you get rid of microorganism, the ecosystem is going to suffer, and that sufferings are going to be more in places like desert, or where primary colonizers are microorganisms. See, once any environment, the first colonizers are going to be microorganisms, and then only you will be able to find annual plant. Then you will have a perennial plant. Then the biome will develop. But if you don't have microorganisms, then the development of all other organisms are going to suffer. And if we get rid of microorganism, well, the function of any biome is also going to suffer. So yes, definitely you will have keystone information about soil microbial communities, water microbial communities, and other systems. But we may not appreciate them till we are at a loss, because if you there is a statement which came from a very famous microbiologist. He said, I don't bother if you don't have a dog, but if they become more concerned, the role of a microorganism and ecosystem function. So if you get rid of one particular organism, maybe like a keystone organism, and then the ecosystem is going to collapse. It will collapse. So keystone, understanding those keystone species, well, it is not going to be just one. That is my guess. It's not just one, few of them, because microorganisms are too many in any ecosystem, because they actually kind of a what you call backup plan. So one doesn't do, the other one will do. So that's why we have so many diverse organisms in an ecosystem. So the diversity is actually taking care about the changes or the hardship they have to undergo. But if you get rid of all those good ones, yes, we are going to have a big problem.
Sir, is there any microbiome related to hormones in human?
Related to?
Hormones in humans.
Okay, I have no idea about hormones. Well, I don't know. Yes, maybe the questioner, the person who asked the question has drawn the parallel with the phytohormones and humans. Well, I don't have any information. So, phytohormones, yes, but hormones, I don't know.
Sir, are there any method or techniques available to communicate effectively with the select microbes in order to understand its nature in comparison with others?
The question is how to communicate with the microorganism.
Yeah.
We have to first understand how do they communicate among themselves. So there are some communication abilities. Most of the communication among the microorganism happen through chemical signals. So what they call is, there are some signals like, what do you call, quorum sensing molecules, and they are responsible for bringing microorganisms together. And there are quorum quenching molecules which will actually get rid of some other molecule. You can communicate a particular group of organisms. So antibiotics is a natural way of keeping off certain group of organisms. We are now making use of antibiotics to keep certain group of organisms, like gram-positive, gram-negative group of organism, also like fungal organism from the bacterial organism. It's possible using antimicrobial, antibiotics, or it is also possible to use antimicrobial metabolites. Possible, right? It is possible.
Sir, can organisms selectively recruit their microbiome?
Yes, they do. They really do, because by giving out a select group of chemicals, in fact, like in the case of plant system, they interact with the microorganism by root exudation. So plants will decide what is the level of abundance or group of organisms they need in the rhizosphere. So they give out signals by root exudation, and then they recruit the organisms. So that is why we have a selection influenced by the plant on the roots. Otherwise, if you look at the soil has got every other organism. All the organisms are present in the soil, whether it is associated with the living organism or with the dead organisms. So both decomposition and living process are all being going on in the soil with the plants. Like in fact, microorganisms present in the soil allow the plants to grow. At the same time, if the plants do not grow, they are the ones which will attack the plants and decompose them. So when the plants become dead, and they will completely mineralize the plant and get ready for the next plant. So they will allow the plants to grow. So that during that process, plants will select the organism which are to be associated to come in the rhizosphere or in the philosphere to a larger extent. And then they will, their association is going to help. So recruitment is going on. Recruitment is very much going on.
Sir, another question. Sir, recently I have studied about a bacteria that consumes electricity, Shevonella species. What makes them to do so?
Because as I give you some introduction, I mean, like I introduced that concept of redox energy, redox couple, and most of the energy which is gained by microorganism is through the exchange of electrons between the, electron donors and electron acceptors. So, and there are organisms which will actually, like Shevonella and the Geobacter, which was actually discovered by Derek Lowley a year before Shevonella was discovered by Kenneth Nelson. They are able to give out the electrons. So that electrons is what we call them as electricity producing organism. So when we have a cathode or anode, and this microorganism can, give out the electrons, and that is all going on in the nature. So we call this group of organisms as electrogens, or and what the whole process is known as electrosynthesis. And this electricity can also be made use of. And in fact, like we understand that soil has got the electrical potential. The same year when scientists also reported the ECG in the human system. So electrical signals which we create by heart pumping, then led to the development of ECG, electrocardiograph, and that is being used to understand about the human health, human heart health. The same year only, Potter also explained about the electrical potential of soil, and this is nothing but all mediated by microorganisms, and those are like iron-reducing bacteria, like Geobacter and Shevonella. They can give out electrons. Yes.
Okay. Sir, is there any possibility to change the behavior of an organism by changing the gut microbes? Will it have any prospects in changing one's adaptation to stress?
I'm not able to, I'll give you an example, probably yes, because there is a disease called autism. That is a kind of a poor development of brain. And right now people are able to understand that, like if you make changes in the gut by giving certain substrates and also using certain microorganisms, it is also possible to get rid of those behavior or the behavioral changes. Okay, that's possible. The other one is if a person is suffering from obesity and not able to control the body weight or body biomass, and if you are able to make a change by fecal transplantation therapy, where a microbiome of a normal healthy human being is transplanted into an obese person, then the person's obesity can be now altered to a normal healthy individual. It's possible. So microbiome applications were actually shown to the world as a possibility, has been actually shown to the world by fecal transplantation therapy. Yes, it is possible to change the behavior and change the behavior and phenotype. I'm just giving you these two examples, but behavior is it's a difficult to decide. In a sense, it's not going to be an immediate effect, but in the long term, one can make changes. Yes.
Sir, genetic engineering of Bt gene application in crops is very successful. Similarly, any other significant gene genetic engineering in crops is expected shortly?
Bt is one, the other one is herbicide tolerance. So we have several maize cultivars with the herbicide tolerance. So it is available. So herbicide tolerance genes are actually from from bacterium, and then we have several cultivars which can now tolerant to herbicides. It's all available now.
Okay, sir. Sir, can you please update us on possibility and progress in nano biofertilizer application?
Well, the talk is not about nano fertilizers because, but of course there are several reports or several works are going on in using nano fertilizers because this can be easily absorbed by the plants in quantities which need not to be large enough like what you do it in normal fertilizers. And there are developments, of course. You can see some of the works of, from TNAU, also from IARI, where they have introduced nano fertilizers related to zinc and then iron, which is now being recommended to the farmers. Yes.
Sir, is there any beneficial virus which can contribute to plants?
It's several, viruses are, not all viruses are bad, some viruses are good, but I don't have an example to say, but maybe one I can say that there are few ornamental plants which is, they have a, different kind of color streaks in them, like tulip, and then they have, some of them having a desirable color streak, which are all because of the involvement of plant viruses. And well, that's one example which I could think of, which is beneficial. But otherwise, most of the viruses are considered to be like disease causing, or like Panchita banana, and then several other viral diseases which are unpleasant, not good for the plant productivity. But from aesthetic look, yes, tulip with some streaks of which are caused by the viral particles, viruses, which are good sense, they are considered to be aesthetically beneficial. Yes.
Okay, sir. So still we are having many questions, lot of questions. I kindly request the participants to ask the questions in the feedback form. So with this, I take this opportunity to thank our guest speaker, Dr. B Ramakrishnan, for spending his valuable time in the midst of his busy schedule and explaining about microbiomes, and he explained about different aspects of microbiology also. So thank you, sir, on behalf of Kumaraguru institutions. I wholeheartedly thank you for accepting our invitation and for delivering the lecture. And I thank Dr. Inya Kumar, Assistant Professor Agricultural Microbiology, for arranging this wonderful lecture for the benefit of our students.